Today in this lecture, we will talk about the extracellular fluid osmolarity or plasma osmolarity and sodium concentration. So far in lectures about a renal system and urine formation, we have discussed so many times that the kidneys can decide to make concentrated or dilute urine. Kidneys decide to make concentrated or dilute urine, but it depends upon the concentration of plasma. Now, one very important thing to discuss is the osmolarity. And osmolarity is actually due to the different type of ions that are found in the plasma. Normally, the osmolarity of plasma or the extracellular fluid osmolarity is around 300 milliosmol per liter. And when we average it out for the internal interactions, it is around 282 internal interactions between the ions. It is around 282 milliosmol per liter. Now, this is the normal osmolarity of plasma, and increase or decrease in the osmolarity basically shows the increase or decrease in concentration of different ions in the plasma. But another very important thing is that around 94% of this osmolarity is dependent upon concentration of sodium only. So sodium is a major cation of the plasma, and around 94% of this osmolarity, the extracellular fluid osmolarity, is dependent on sodium concentration. The remaining 3 to 5% is dependent upon other solutes, which may include glucose or urea. Now, sodium and its anion are the major contributors. So the regulation of sodium, by the kidneys is very much important for the regulation of plasma osmolarity or extracellular fluid osmolarity, because this thing is ultimately going to determine the formation of concentrated or dilute urine, which means whether the body is going to conserve water or throw out extra water in the form of urine. Now, normally the concentration of sodium in the human plasma or the human blood is around 140 to 145 milli equivalent, and the average is around 142 milli equivalent per liter. To calculate the osmolarity easily, we normally use an equation which is 2.1 into 142. 2.1 into concentration of sodium in plasma. It will lead to an average osmolarity of plasma. So the formation of concentration in dilute urine is dependent upon the osmolarity of plasma, and 94% of osmolarity of plasma is dependent upon the concentration of sodium only. So we can roughly calculate the plasma osmolarity just by calculating the sodium concentration and putting that in this equation, which normally would give us around 298 milliosmol per liter. Now, in some circumstances, other solutes like glucose and urea also become very important. To calculate in the exact osmolarity in those circumstances, we calculate it by the equation 2 into concentration of sodium plus concentration of glucose plus concentration of urea. That will give us the plasma osmolarity in those circumstances. Now, why is it important to discuss so many times? Because ultimately, any change in the concentration of sodium is going to affect the urine formation process, and which has been the main topic we have been discussing so far. So to regulate the concentration of sodium in plasma, we normally have two major mechanisms. One is the ADH osmolarity. One is the osmoreceptor ADH system, and the other is thirst mechanism. Now these two mechanisms, the osmoreceptor ADH system and the thirst mechanism, will try to maintain the concentration of sodium in plasma, and then the concentration of plasma is going to maintain the plasma osmolarity in check, and the concentration of plasma or the osmolarity of plasma, when maintained normally, will lead to normal formation of urine, and or it will affect the urine formation process. So that's why it is important to regulate the concentration of sodium. Now, in the coming lectures, we will be discussing in detail the osmoreceptor ADH system as well as the thirst mechanism in detail. Thanks a lot for watching the video.